Good morning. What we do is deliver antibodies to intracellular targets that are commonly called undruggable because they are very difficult to reach, especially for antibodies. Our work is focused currently on cancer, but there is a large majority of diseases that stem from dysfunctions inside the cells. Some people say 75%. They can be addressed with small molecules, but many of these small molecules go over the body and they may be toxic and have side effects. Antibodies would be ideal because they are very specific, but they cannot enter the cells themselves. So what we do is actually we help these antibodies enter the cells. Our work stems from originally the work of Professor Alonso with Bob Langer at MIT. About 25 years ago, Professor Alonso worked on all kinds of deliveries. If I have any question about LNPs or whatever, she always has an answer. And she has a lot of publications. She's a star in the world of nano delivery and delivery in general. We have a few oncologists also on board in Spain and in the US that help us sometimes even run their own experiments on our products to confirm what we see. I'm Olivier, Jarry, CEO. I'm coming mostly from large pharma, and our COO, Dr. Terrejo, is from academic and nano delivery as well. So how does it work? Overall, it's a nano capsule made of polymer. We call it a multifunctional polymeric nano capsule. It's a bit flexible actually, and can squeeze into the vasculature of a tumor. From the outside is just a bubble of polymer. It's neutral, it circulates in the blood flow for weeks without any degradation before it goes to its target, the cancer cells. Once it reaches the target, it's the surface of the polymeric nanocapsule melts with the membrane. There is endocytosis, endosomal capture and release, and then the antibody is released to its destination, and we'll see a few images later. The antibodies inside in green are in water phase. They are not conjugating, they are not transformed, they are not engineered, they are not modified, they are really the pure antibody, IgG1 typically, but we can do different sorts. We can do also bispecifics, fragments, we can do different things, but they are really not modified, meaning that when they are released, they are just active and go to their targets. You see in the center in yellow a drop of lipid. This is actually for structure. It's the way that actually we build the nanocapsule, the antibody attached to the lipid, and then we form the polymer around the antibody. But we can also incorporate a small molecule inside. And finally, on the surface of the polymer, we can attach ligands. And these ligands are of various types. Right now we are using ligands that target cancer cells, but we can target other types of cells as well. Actually, the next slide gives you an idea about the different options for functionalizing these nanocapsules. On the left, targeting first. So even if we use nothing, we use very commonly hyaluronic acid as a polymer. It has an affinity for CD44 receptors on cancer cells. We have been using also a peptide called TLIP1, and has an affinity for NRP1 and NRP2 receptors on cancer cells in humans, in dogs, in other animals. We are looking at the next generation that actually has an affinity for the vasculature of the tumor as well. And we can use antibody fragments, we can attach different things on the polymer, not on the antibody, on the polymer. In terms of a cargo, we can have either a biologic in the water phase. Today we have developed two antibodies, proprietary antibodies for KRAS mutation G12D and G12V. And we are working on the next four antibodies, just starting, STAT3, CRAF, HIF1 alpha, HIF2 alpha. We can also deliver mRNA, siRNA, bispecific, and other compounds in the water phase. In the lipid phase, we can deliver small molecules. So we are working with our own small molecule. We deliver generics like docetaxel, paclitaxel. These I'm working to license them to generic manufacturers, but we are also working with a partner on their PROTACs to deliver PROTACs directly into the cytosol. And then we can have a combination of both, because they are in two different phases. Right now we are testing our KRAS antibody with a MEK inhibitor small molecule in order to attack the MAPK pathway in two different locations. What are the benefits? A full page here. I'll try to not cover everything, but manufacturing is actually very simple. Looks complicated, it's actually very simple. It's two mixes with the different ingredients. Everything that we use which is non-active is today FDA EMA approved and considered to be very safe, like hyaluronic acid, like triglycerides. We use no solvents. We use no high temperature process which could degrade the antibody, contrary to many other nanocarriers. We use doses that are very moderate, 10-15 milligram per milliliter. So this is typical of antibody use in general. If you think of other constructs, like we'll see on next page, many of the other constructs use a large number of antibodies because a lot is lost. Here, because we protect them inside the polymer bubble, essentially we have very few losses and we can use a relatively low dose. It's very stable in storage. We have 6 months now, very good stability at 4 Celsius. We are looking in a couple of weeks at 12 months, and we believe that it's very stable for 12 months. And obviously very stable at lower temperatures. Biodistribution, a few things to note here. These nanocapsules are very stable in plasma. So I mentioned no degradation. For weeks and weeks they can be circulating. They are not like LNPs sent to the liver in a couple of hours, at least the LNPs I've tested. They just stay in plasma. Depending on the ligand that we attach, obviously they can go more or less quickly to their targets, to the targeted cells. All of this is because they don't accumulate a protein corona. So if you worked on nano delivery, if you work on medical devices, you know that very quickly they may attract proteins that attach on the surface and essentially prevent the device or the molecule to work. In our case, and even antibodies themselves that attach a lot of proteins. In our case, after days and days, we have virtually no protein attached to the polymer, which explains the fact that it keeps circulating. Also very interesting for some applications, we found a very high diffusivity. We tested only the brain because it's difficult and the liver because it's easy. But in the brain, if you inject LNPs or you inject directly an antibody, it just stays where you inject it. There is no real movement around. If you inject these nanocapsules, they start diffusing into the entire CSF and make over the entire brain, which has a lot of benefits, obviously. In the liver, we have some pictures, not here, but we can actually diffuse in the entire liver to treat the liver as well. And finally, safety, as I mentioned, it's all safe ingredients or ingredients known as safe, and the antibodies are very specific, so they have no activity outside their target. Very different from antibody-drug conjugates. Sorry for everyone who is excited with ADCs. They may be great in some cases, but the antibody is the ligand or is the targeting moiety, not the active. The opposite is drug-antibody conjugates, where the antibody is a payload, but the drug is actually the targeting ligand. There are some entities working on this. I've heard they are not great. They are difficult to handle. The antibody is not protected, so that's one of the cases where you have degradation and a lot of loss, and you have to use bigger quantities. And very different from other constructs, nanocarriers like LNPs or liposome. These cannot really load antibodies. They are not designed for it. LNPs are good for mRNA. You can get maybe a couple of antibodies inside. It's difficult and it's not efficient. So our first two assets are KRAS antibodies. KRAS is very well known. Each of these mutations is in more or less 1 in 4 pancreatic cancers and a long list of other cancers. We do I have a number of proof of efficacy. We see that 100% of the cells receive the antibody within 24 hours. We see the antibody migrating to the target. In the case of KRAS, it's on the inner surface of the membrane. And we see this is only after 8 hours. But we see on the right, the engagement with the target keeps increasing after 24, 48, 72 hours as the nanocapsules are reaching the cell. We have a list of POC studies. This one is a relatively difficult pancreatic cancer where we see we are better than the standard of care. We have a poster here just at the outside which has another graph with an even more difficult pancreatic cancer where we control the tumor growth completely when the standard of care is essentially not working in humans, not working in mice. Our business model is to develop our own antibodies and carry them with a vehicle, but we can also carry the antibodies of other people, or mRNA, or siRNA, or others. We have today collaborations, 2 in Canada, 2 in the US, 2 in the UK, actually it's by twos, and 2 in Belgium, 1 in France, 1 in Spain. And we are very happy to take new collaborations and deliver the products of somebody else. We started with our own cancer targets. Now we partner antibodies for other cancer targets with other companies. We have other modalities, as I mentioned, and we are going into actually neurology, virology, hepatology, inflammation, which is reflected here on our pipeline. You see the 2 KRAS, the STAT3. Actually, we have the name of the partners here. These are partners just for developing the antibody. They are not going to commercialize, so this will be available for commercialization. And we expand the platform into virology, neurology, PROTACs in terms of modality with a partner here, and mRNA. Patents are covering the technology to deliver antibodies. There are some specifics, and then every antibody that we develop. So this is it. Taking any question?
All right, thank you so much. Do we have any questions?
Hi, I'm Vera for NRW Venture, and I was wondering if you could say a couple of more sentences on the business model. Specifically, are you also looking to develop your own products, or
Yes.
are you only interested in out-licensing?
No. The products that you see before, all of these are own products.
Because it says on the right available to out-license, so I was wondering.
They will be out-licensed to Big Pharma. So the model is to develop as far as we can and then license to Big Pharma. So now with little resources, if we can outsource at IND time, that will be great. If it is phase 1-2, we'll have to push through phase 1-2. We believe once we build credibility, we can outsource earlier. Right now, the collaboration that we have are either supported by grants or by the partner, and it's a small income that covers the cost and more. But this is just for the launch. As we move forward, we will have more and more of our own products.
Thank you.
Have you got any more questions? Yep.
Hello, I'm Emily Said from Parkwalk Advisors. Could you say something about the immunogenicity of the
Sure.
Yeah, of the MNP? Because you mentioned that about your competitors already.
Yeah. So all the ingredients, hyaluronic acid, triglycerides, other polymers that we use, other fat, the water, all of it has zero immunogenicity. So if we see no sign of toxicity in mice, there is no reason why this would be toxic. So only the active ingredient can be toxic. Most of the antibodies that we use have very, very specific targets. So if we use this antibody, they have no activity on the wild type, and therefore they go only to the mutated cells. Some targets have other functions in the body. A big pharma asks us to work on ERK1, ERK2. This is dangerous because it's involved in many other mechanisms. So we said no. We don't want to do that because we believe it's quite unsafe. But otherwise, there is no immunogenicity. When we deliver a small molecule, yes, it can be a cytotoxic like docetaxel. But in that case, we are very targeted. So in one experiment on docetaxel, we increase the concentration by 40 times in the cell compared to just an injection. So even when handling something toxic, we reduce toxicity.
All right, thank you very much, Olivia. I believe that's all we've got time for.
And there is more on the poster outside.